Hello everyone, in this video we will be discussing the fundamental principles of colorimetry, including the laws of colorimetry as well as the basic structure and function of a colorimeter. We will also explore how colorimeter calculates optical density and how this information can be used to estimate various parameters. We will also demonstrate the establishment of Beer's law in laboratory with graphical representation. And last, applications of colorimetry. We'll start with introduction. What is colorimetry? Colorimetry refers to measurement of intensity of color. What is colorimetry? Colorimetry refers to measurement of intensity of color. It is used for measurement of colored compound or converted into colored compound by suitable reaction. In biochemistry, we are doing this second part, converted into colored compound by suitable reaction. Suppose we want to perform estimation of plasma glucose. So what we do is we convert plasma glucose targetedly into colored compound. Okay. Then we check the intensity of that color. Next is, what is the advantage of this method? It is very sensitive method and requires very less amount of sample. Okay, you must have seen that laboratory person takes few ml of blood sample and gives results of so many parameters. So in biochemistry we are performing all the estimation based on this colorimetry. Okay. Suppose after suitable reaction we convert all the plasma glucose into colored compound and we get this kind of color. Now we have to check the intensity of color. So what we do, we will pass one light through this solution. Okay, light before enters into the solution, it is called incident light. Whatever the light pass through the solution, it is called emergent light. Okay. So whenever the light passes through any colored object or colored solution, some of the light will be absorbed and some of the light will be passed through the solution. Okay. Some of the light will be transmitted through the solution and some of the light will be absorbed. So whatever the light will be transmitted, it is called transmittance. It is called transmittance T. And theoretically it is emergent light upon incident light. Emergent light upon incident light, that is transmittance, light pass through the solution. Now if we do reverse thing, we will get absorbance. The light, whatever the light will be absorbed. If we reverse this transmittance, we get absorbance. So theoretically it is log of 1 upon T. I know this is very confusing, but pay attention, it is very easy. Just remember, whenever any light passes through the solution, some of the light will be absorbed and some of the light will be transmitted through the solution. Okay. Now this absorbance depends upon three things. Number one is color of the solution, means intensity of color. Next is pathway of light, and third thing is wavelength of light. Okay, so absorbance, whatever the light which is absorbed or transmit, whatever the light is absorbed, that depends on three things. Number one, color of the solution. Number two, pathway of light. This is pathway of light. Okay, this is pathway of light. And third thing is wavelength of light. Now for some time keep this wavelength aside. Number one, color of the solution and this pathway of light. Depending on this, we have two laws. Number one is Beer's law and second is Lambert's law. Beer's law and Lambert's law. So there are two laws. Number one is Beer's law. Number two is Lambert's law. What Beer's law says? Absorbance is directly proportional to the intensity of color when the pathway of light is constant. What does it mean? Suppose we have three test tubes. Okay, Suppose we have 3 test tube. Color of this first sample is little lighter, then number second is more intense, and number third is more than intense than this second one. So absorbance is directly proportional to the intensity of color. So pathway of light is constant. Here pathway of light is constant. So absorbance is directly proportional to the intensity of this color. So absorbance will be highest in this test tube, then this, and least will be in this. Okay, understand, pathway of light is constant, so the absorbance will be lesser in this and maximum in this solution, because the intensity of color is more in a third test tube. Okay, now what is Lambert's law? Lambert's law says that absorbance is directly proportional to the pathway of light when the intensity of color is constant. So suppose we have one solution, then we check the intensity, we pass the light, and the pathway of light is increasing. Okay, so when the pathway of the color is same, color is same, but the pathway of light is increasing. Pathway of light is increasing. So absorbance will be maximum in this third and least in this first one. Now check the intensity of color. We use this instrument. This instrument is called colorimeter. This instrument is called colorimeter. Now how colorimeter check the intensity of color? So we need to understand the structure of colorimeter. So this is structure of colorimeter. What is inside the colorimeter? So we have to pass light through the solution. So we need one light source, that is bulb. Second thing is lens that converts the light. Then light passes through this slit. Then there is monochromator. This is monochromator. After passing through this monochromator, again comes from the exit slit. Then pass through the colored solution. After passes through the colored solution, light will be reflected on this photodetector. Photodetector is made up of selenium, either gold, silver. This photodetector will generate electromagnetic wave, and this readout device will give one reading that is called optical density. It is called OD. Short form is OD. Okay. Now understand the importance of wavelength. Importance of wavelength and the role of this monochromator. Importance of wavelength and what is the role of this monochromator. Now see this table. Now understand the importance of wavelength. We will pass that wavelength of light which has maximum absorbance in a particular colored solution. Okay, suppose we have red color solution, so we will keep the color of filter paper, that is monochromator. Color of this monochromator is nothing but colored gelatin paper or colored glass. Okay, so if the color of the solution is red, then we keep the bluish green filter paper, bluish green monochromator. So the 505 nanometer of light will be passed through this solution. Okay, 505 will pass through this solution. So this table shows that which color is developed and which color filtered or monochromator we will keep. So which wavelength of light will be passed. So we will pass, again I am telling you, we will pass that wavelength of light which has maximum absorbance in a particular colored solution. Now again understand this slide. We have this test tube and we need to check the intensity of this test tube. Okay, intensity of this, color of this test tube. So what we do is we will take one test tube with distilled water. Okay, and we'll press this button set 0. Okay, set 0 means whatever the light that comes through this distilled water onto the photodetector, it is 100%. Okay, it is 100%. Whatever the light comes through this distilled water, it is 100%, and transmittance is 100%. So absorbance is 0. Okay, light comes through this distilled water is 100%, transmittance is 100%, absorbance is 0. Okay, by pressing 0 we have set transmittance into 100%, so it is in a percentage. So absorbance is also in a percentage, so we can write 100 here because it is in a percentage, so we can write 100. Okay, so log of 100 minus log of t, we can write. Okay, log of 100 minus log of t. Okay, after putting this colored solution, colored test tube, suppose the transmittance is 70%. Suppose transmittance is 70%. So log of 100 minus log of 70. Okay, log of 100 is 2 and log of 70 is 1.84. So we get 0.16. 0.16. So in readout device we get one digit, that is 0.16. Okay, now got the point how absorbance is calculated by this colorimeter, and we get one number, and this number is called optical density. Short form is OD. Okay, so till now what we have is one digit, that is optical density. Okay, optical density of this colored compound, optical density of this unknown test solution, and we have to measure the plasma glucose level, and we have converted this plasma glucose targetedly into colored compound, and we have one number. Okay, till now we have one number, one OD of test solution. Now can anyone tell me how strong this person is? So to tell how strong this person is, we need one reference person. Okay, we need one standard to compare. Okay, so after comparison we can say this person is stronger than this person. Okay, we need one standard person. Okay, we need some reference. Okay, so what we do is we will make one standard solution. Suppose we are going to estimate plasma glucose level, so we will make standard of 100 milligram per deciliter. We will make standard of 100 milligram per deciliter. Okay, this is unknown solution, this is test solution. We have OD of this test solution, that is 0.16. Okay, 0.16. Now we have standard solution, we have this concentration of this standard solution, that is known concentration 100 milligram per deciliter. Okay, so till now what we did is we took two test tubes, we add standard solution in one test tube, test solution in other test tube, then we have added reagents, and convert this glucose, plasma glucose, into colored compound in both the test tubes. Okay, in standard as well as test solution. After the incubation we get 0.16 optical density of this test solution. Suppose we get OD of standard is 0.12. So we have three things: standard concentration, OD of test, and OD of standard. So we have three things: OD of standard, that is 0.12, standard concentration, that is 100 milligram per deciliter. Then we have OD of test, that is 0.16. So how much the concentration of test? So concentration of test is OD of test into standard concentration. Okay, OD of test into standard concentration divided by OD of standard. OD of standard. Okay, simple. OD of test into standard concentration divided by OD of standard. Okay, 0.16 divided by 0.12 into 100. So here answer will be 133.33 milligram per deciliter. Okay. So concentration of this test solution is 133 milligram per deciliter. Now you can see this, I have deliberately left this space. Okay. So we don't take 2 test tubes, instead we take 3 test tubes. Third test tube is blank. Third test tube is blank. In standard we add standard solution. In test marked test tube we take test solution, and in blank we take distilled water. In all three we add reagent. We add reagent in blank also. Now colorimetry is all about color. We need exclusive color of this plasma glucose which we have get from suitable reaction. Okay. Now if there is a color of reagent also, it will interfere in this OD. So what we do is we add reagent in blank and take OD of this blank, OD of this blank solution, and will minus the OD of blank from test and from standard also. Okay, we don't need the color of reagent. Understand what is the importance of blank? Importance of blank is to minus the color of reagent. Okay, so final formula will be concentration of unknown solution test solution is OD of test minus OD of blank upon OD of standard minus OD of blank into standard concentration. Okay. In exams, many students make mistake in putting this formula. They put this standard in numerator and test in denominator. So the result will be opposite. So always have habit of checking the color of test and standard. If the color of the test is more, then the result will be more than standard concentration. And if the test color is less than the standard, then the concentration will be less than the standard concentration. Always keep in this mind. Okay. Now we'll go into the practical lab and we'll establish the Beer's law. Okay. So let's go.
So this is colorimeter. You can see here the list of filters and its wavelengths. With this dial, you can change the filter. Now we will start the instrument. These are the test tube with potassium permanganate solution of increasing color intensity. Now first we will put the test tube with distilled water in colorimeter and will press 0, means we'll set transmittance as 100% and absorbance 0%. Now we will see OD of all the colored test tubes one by one. First sample has OD of 10. OD of second is 20. OD of third is 34. Fourth one has OD of 43. And last sample has OD of 53. Now if we make a graph of these values, we can see the line is almost at 45 degree. Means OD is directly proportional to the intensity of color. Now lastly, we will see applications of colorimetry. It is widely used in labs for estimation of biochemical components in various biological samples like blood, plasma, serum, CSF, urine, and other body fluids. Routinely, colorimetry is used for estimation of glucose, urea, creatinine, uric acid, bilirubin, lipids, total protein, albumin, enzymes like SGOT, SGPT, alkaline phosphatase, minerals like calcium, phosphorus, etc. So this is all about colorimetry. Hope you find this video informative. If you do, then please like and share this video and also subscribe my channel. Thank you.